Hello, anatomy students. This is for those of you who missed the lecture due to the MEA science augmentation, or if you just feel like you need a little bit more time on this because I realize that it is complicated. So this lecture is about sliding filament theory, and the corresponding pages in the book, if you would like to follow along or spend a little more time reading about it, are pages 190, 191, and 192 in our textbook. At this point, take out your guided notes that you already have, and I will tell you what to write as you go along. Okay, just a quick review of the structures that we're working with here. This is that connective tissue, the myomycium, that wraps around each muscle cell. Within the cell, we have this structure that's yellow here, where we're going to get initiated here, and that is the sarcoplasmic reticulum. So that's the first thing that's going to come up. Anything else I want to tell you about? There should also be a sarcolemma here, which isn't really shown, so maybe this isn't a great image, but the sarcolemma is the cell membrane. So you would have the myomycium, and then inside of that, sarcolemma, and then inside of that, the sarcoplasmic reticulum, which contains a bunch of calcium that we are now going to utilize. Okay, now we are in the muscle cells looking at the very fibers. We're looking at a sarcomere. So from here to here, Z-line to Z-line, that is a sarcomere. And you can see that this is in the relaxed position, and this one is in the contracted position. And the main difference you can see is that the H zone, has completely disappeared, and also these regions near the Z line have shortened. So we're going to be looking at what's actually happening to cause that shortening, which isn't really shown in this contracted version. And I'm just going to walk you through that. Okay, step one. Action potential, which we've already established because we had the neuron receiving the stimulus from the brain, and that neurological signal passed down the axon to the axon terminals and initiated a whole series of chemical events that eventually leads to action potential within the muscle cells. So once that's happened, action potential stimulates the sarcoplasmic reticulum to release calcium ions into the muscle cell cytoplasm. At this point, a couple of things start to happen. One, we get ATP kind of pulling these myosin heads back into position. So these will go this way, these other ones will go this way, and that zone here that's going to get contracted is in the middle. So we use ATP, and then the other thing we use is calcium ions. So these calcium ions come in and they actually attach to these regulatory proteins. And when we attach any molecule to a regulatory protein, it changes shape. Once that shape change has happened, this particular little piece here, which is essentially blocking the myosin head from attaching to that actin, allows the myosin head, which has a very strong affinity for that site, to attach to the actin. Once that attachment occurs, actually I think it's on the next page, so I'm going to show it to you. Okay, I got a little bit ahead of myself. So there's the calcium floating around inside of the cytoplasm. Calcium attaches to these regulatory proteins, which moves them along the actin and opens up the binding sites for myosin to bind to actin. Once that binding occurs, the myosin heads pull, depending on which direction they were sort of loaded, pull towards the middle, kind of like pulling on a rope, like hand over fist, if your two separate hands were different myosin heads. So each place where your hand would attach to a rope, that would be an attachment site for myosin on the actin filament. Once they've pulled together, this space decreases in size, the entire muscle decreases in length, and when we contract something, we're decreasing the length of that muscle. So essentially, these filaments slide past each other, which is why this is called the sliding filament theory. Here it is all together. We have step 1, nothing has happened yet. Step 2, we add the calcium. This doesn't actually show the myosin kind of getting pulled into position by ATP, but it would move position when it is loaded with ATP. And then grabbing onto the actin and going back to its usual position and pulling the muscle fiber together. Another thing I would love to see in this diagram, rather than just seeing this space decrease in size, is the whole length of the muscle fiber decreasing. And then here's that same diagram from the very beginning. You can see the Z line here and the Z line here, and this is reduced in size, this is reduced in size. This muscle is contracted. And I provided a video here that actually comes with our textbook. Feel free to give it a look because it's a much better illustration of what I was able to do with the stylus here and the static images. I'm hoping this will work when I export it.